Hello there, my name is Redeat Tibebu. I'm a PhD candidate at the Board of Lab at the University of Minnesota. Before I start, I want to say thank you for the organizers of this NAPB conference, especially Sarah Davison for allowing me to talk to you a little bit about my interest in using advanced plant biotechnology tools for improving orphan crops and what I'm calling green revolution to gene revolution in orphan crops. So just to give you a little personal background and my connection to this topic, I grew up in Ethiopia, which is a country in sub-Saharan Africa, and a country that's heavily reliant on agriculture for both domestic use and export. However, Ethiopia suffers from drastic food shortage and malnutrition, and there are multiple factors for this, but I want to focus more on what I believe is a major solution for agricultural productivity, not only in the country, but also in sub-Saharan Africa, where climate change is expected to exacerbate the situation, the current situation, and where it's expected that population will double by 2050. And so I think it's a smart way to improve the crops that are cultivated in these countries by using advanced plant biotechnology tools coupled with traditional breeding. So the Green Revolution, you all know, brought about drastic food security improvement, agricultural productivity in the regions that it was implemented and where it was successful. And however, Africa did not benefit from this. The Green Revolution was not successful for the most part, and this was because of the fact that Africa has very diverse climates and crops that are cultivated throughout the continent, which makes it very difficult for traditional breeding. And most of these countries did not have the investment power to build the pipeline for improving these crops through breeding. Some of these crops that are cultivated throughout sub-Saharan Africa are shown here. Sorghum actually isn't an orphan crop anymore because of the recent improvement work that's been conducted on it, increasing the yield and the adaptation to various abiotic stresses that it used to face. However, most of these crops have well adaptation to the regions that they're grown in, which tends to be harsh climates with water scarcity and also various abiotic and biotic factors. Also, some of these crops tend to have pretty good nutritional benefits in terms of having some nutrients that aren't found in major crops. For example, teff is a cereal crop that's cultivated in Ethiopia, and I'll talk about it later on. But it is a cereal grain with high calcium and iron, and it's also gluten-free. So it's actually gaining popularity now in the functional or gluten-free food markets because of this characteristic. However, though, even though these crops have all these benefits, both in agronomic and nutritional sense, they tend to be very low in productivity and labor intensive for cultivation and also post-harvest processing. And this is based on mainly because of the fact that they haven't had much improvement other than the selection process that farmers do when planting these crops. And so hopefully with the advent of gene editing tools, we can apply these techniques to improve these crops. And when we talk about gene editing tools, I want to make sure that everyone is on the same page. So gene editing allows for precise targeted modification of genomes. We're talking about plant genomes here. However, it does require genomic resource data or reference genomes and the knowledge of genes and their functions, which traditional breeding doesn't really require, right? but it is more rapid and more precise if you have those data and those resources. There are various tools available currently for specific applications, such as insertions and deletions using talents or CRISPR-Cas9. And then base editors are the most recently reported, which allow for specific modifications of base pairs or amino acids for a more precise and alteration of the gene expression rather than deletion or insertion. So there have been various applications of these gene editing tools in various major crops as shown here, and the application ranges from benefits such as resistance to abiotic stress factors, biotic stress factors, overall yield, as well as consumer benefits. So I was involved in a project that improved potato storability by just targeting one gene using talents. And so we were able to knock out this gene that controls the reduction of sugars, which when they are stored and processed at high temperature, for example, when making fries or chips, these reduced sugar tend to turn into these bitter brown spots that have been reported to have acrylamide, which is a possible carcinogen. So by just knocking out this one gene, we're able to produce potato lines that are healthier to consumers. So moving these gene editing approaches to orphan crops require various advancements in technology, such as sequencing, phenotyping, and bioinformatics. And there has been rapid improvements in these technologies. Reference genomes for orphan crops and for different varieties have been reported and are being put into public accessible databases, which will help researchers globally to be able to access these and perform their own gene editing applications in the crops that they're interested in. And also these advancements are helping improve the gene identification and functional characterization work. And so it takes less time than it used to take with some of with the utilization of these tools. Another advancement that's key is advancements in plant transformation methods. You might have heard that plant transformation tends to be the bottleneck for gene editing, tissue culture specifically, delivery of the editing reagents and then into the plant tissue, and then going through tissue culture to try to get regenerate a transformed plant usually takes a lot of time, expertise, and resources that most labs in especially developing countries don't have the capacity for. And so genotype independent transformation methods using developmental regulators, which has been really at the forefront of this field, is helping move forward this diminish this bottleneck. And Cassava Agrosciences has done a lot of work in this forefront, and also sharing their techniques and technologies with institutions and researchers globally. Another one is plant tissue culture-free transformation methods. Our lab at the Dan Voitas lab reported 2 papers last year where one used virus to transform or edit plants, and then another one used agrobacterium to also edit various dicot species. And both of these methods skipped the tissue culture process, which not only saved a lot of time, but resources as well. And so some of these advancements are key Of course, communication and collaborative platforms are also have grown through this COVID time. Zoom and other platforms allow for data sharing and working together for training purposes. And so this allows for the sharing of these tools and techniques with researchers that may not have had access to it in previous years. There are various trait improvements that can be carried out in orphan crops. As you can see here, some of these orphan crops suffer from these biotic and abiotic factors, and there are various genes that have already been characterized to improve some of these traits. And so plant architecture, for example, most of the cereal orphan crops tend to suffer from plant lodging, which was a huge issue before the Green Revolution in both wheat and rice. And these genes, the SD1 and RHP1, were introduced through the traditional breeding that took place during the Green Revolution. And these could easily be introduced in orphan crops, orphan cereal crops through gene editing, as well as some of these other traits and genes associated with them. And so these are very low-hanging fruits that can easily be introduced in various orphan crops in order to reach their yield potential and improve their nutrition as well. One case study I want to talk about is cassava. Cassava is the most important food crop globally, and also it feeds 1/3 of Africa's population. It is well adapted to that environment, but unfortunately, it does require, it's a labor-intensive cultivation process and post-harvest processing. And the first reference genome was reported in 2009, and since then, 2017, the first demonstration of CRISPR-Cas9 was reported, and since then there have been a lot of ongoing work in improving cassava through gene editing approaches. One from our lab in collaboration with the Danforth Institute was developing herbicide-tolerant cassava targeting the EPSPS gene using CRISPR-Cas9. And this glyphosate-tolerant cassava is now being tested, field testing in the countries in sub-Saharan Africa that cultivate cassava. Another example that's dear to my heart is teff, which is a cereal crop that is the staple crop in Ethiopia. I grew up eating it. It covers about 30% of the land that is allocated to cereal production in the country. However, when you look at the yield, it's the lowest yielding of all the cereals that are also cultivated in the country. And this has to do with the various limitations listed here: susceptibility to weeds, plant lodging, and seed quality. The improvement of teff is now just starting, and this was because of the fact that we didn't really have a full, complete genome of teff until recently. Last year, complete genome was reported and made available, and since then there's been ongoing work to try to introduce some of the genes that I talked about previously to reduce some of these limitations and reach the yield potential of teff. So with that, I would like to conclude stating that technology advancements and increased access to these technologies are key to using advanced plant biotechnology tools for orphan crops. And so I talked about availability of genome being key, and also capacity building in developing countries is another key factor in moving this technology forward. So instead of conducting the research of improving these crops in Western countries, it's best if we can build a capacity, train scientists, researchers in developing countries so that they are able to carry out these improvement work on their own staple crops. And hopefully acceptance of these edited products and better policy landscape in these regions can really bring about these advancements to farmers and consumers. Finally, I am hopeful that we will be able to adapt to climate change using these advanced tools by de novo domestication of wild type crops, which has already been reported, and also improving these various orphan crops and different varieties to be able to develop climate resilient agricultural landscapes, and that is also high yielding and that meets the nutritional needs of the growing population. So with that, I would like to thank you for your attention, and I look forward to our discussions later on.